Second session of the morning. So now we're going to start with Katherine. She's going to talk about Arctic skuas ocean scale migration and population level processes. Okay? You know how this works? Thank you.
Hi, welcome back. Thank you very much for the opportunity to present today and to... Closer? Better? Can you hear me at the back? Yeah. That sounds good. Okay. Yes, just say thanks to the organisers for such a great conference. It's been amazing. I'm Kat Snell. I'm now based at the Max Planck Institute of Animal Behaviour. And throughout my career, I've worked on animal migration and interactions, investigating movements with tracking technologies. And today I'm really excited to present a recent body of work on the Arctic skua study system. This seabird breeds across the Arctic and North Atlantic, and much of this work only exists due to a large collaboration of researchers and field workers across this region, supported by their organisations. Some of the work I will showcase has been led by others, so full credit for their efforts in bringing these projects together. I mean, that lies with them. And it's been such a dream to work together and sharing ideas on these studies. And my research is focused on the interaction of three themes: animal movement, environment, and ecological processes, particularly demography. My work explores how movement influences higher order processes of persistence, speciation, and differentiation of phenotypes and distribution, how the environment shapes animal behaviours and interactions, and how this balances with the intrinsic physiological regulatory control of behaviours and energetics. And seasonality intersects all these three themes. So I'll quickly introduce the study system and species group I work with throughout my career. The Arctic skua, or parasitic jaeger. It has a number of really valuable traits which lend them to being excellent candidates as indicator species for the health of the marine environment and also for fundamental research. It has a breeding range throughout the Arctic and subarctic. It breeds in colonies with densities varying relative to the habitats. For example, in Scotland and the Faroe Islands, they're really tight colonies, whereas in northern Norway, they're more dispersed. They breed on open landscapes, and that's got the additional benefit of enabling long-term monitoring of populations and behavioural observations. We can see them. Although much of the population breeds in the high Arctic, so totally inaccessible regions, so there's still many regions where there's little or no data. It's a long-distance migrant, and its strategy can be described as true migration with long-distance, directed seasonal movements between discrete sites. This is in contrast to many of the seabird species we've seen today, which have this dispersive movement out into the open ocean during the non-breeding period. It's a case-selected species. We're at a seabird conference, we know this, and they're an absolute top predator in the marine and terrestrial realms, and a number of the populations are kleptoparasitic. Like many of seabird species, they are of conservation concern, but these trends appear to be population-specific. And furthermore, they're large enough we can instrument them with GLS, GPS, loggers. So I'd like to take you through a number of studies that all have the common theme of population-level processes. So how phenology and carryover effects, migratory strategy and connectivity, the environment and thermal physiology all may influence high-level processes. Long-term ringing data, together with multi-population data sets spanning the species range in the North Atlantic, is essential for answering some of these fundamental questions. I've actually skipped a lot of the analytical methodology, but please check out the papers or just come to me or Nino Hanlon, who's here today. And there's also lots of really cool ecological facets in the publications, so grab me if you want to know more. In 2016, together with Shurahama, we began intensive field season in the Faroe Islands. Now, first we conducted a national census, the first for several decades, and on Fulai we initiated population monitoring, breeding thermal physiology, and movement studies. This included the deployment of GPS tags to investigate foraging energy budgets, and together with a large consortium across the North Atlantic, we deployed and retrieved archival GLS loggers. And this data, as we've seen throughout this conference, gives this unprecedented insights into movement ecology of long-distance migrants. And changes in light level patterns also. It's possible to determine some of the behaviors, such as onset of incubation. The first project investigating the carryover effects of migration was led by Rob van Velmen from the Wageningen Marine Research and Norwegian Institute for Nature Research. And in long-distance migrants, timing of key events is generally tightly scheduled. And unless this is buffered, this variation in optimized schedules can carry over and ultimately impact fitness. So we wanted to investigate the extent to which any phenological carryover effects at key stages during that annual cycle are driven by population, either at the breeding site or the wintering site. So first we needed to know where and when birds migrate and how connected those discrete breeding populations are at their wintering grounds. We used leg-mounted light-level geolocators in order to obtain daily positions and behaviors at 12 sites across their range. And these cover different habitats, latitudes, colony structures, and main prey sources. So from this data, we're able to identify timing of key events in the annual cycle and migratory schedule. So we found that birds use multiple discrete wintering sites across the Atlantic Ocean basin and into the Indian Ocean. And generally these were thousands of kilometers distance from the breeding grounds. And the extensive individual differences in migratory behavior and low migratory connectivity, i.e. like a high degree of mixing between breeding populations during the wintering site. But breeding at higher latitudes was also associated not only with later reproduction and migration, but also faster return spring migration and a shorter time between return to the breeding site and clutch initiation. So as you might expect, migration to more distant areas was associated with more time spent on migration and less time spent at the wintering areas. But remarkably, much of this variation in timing was compensated during that northbound spring migration, resulting in this really tight population-specific synchrony in timing of arrival at the colonies prior to the onset of breeding. So that birds are capable of strongly aligning their schedules immediately prior to the onset of breeding is indicative that there is actually no phenological carryover effect from the wintering to the breeding period. But akin to optimal migration theory, this observed prioritization of timing may incur a trade-off with energetic costs. That northward spring migration in particular and the North Atlantic staging area, up there in yellow, rather than that wintering area, may influence productivity and hence differences between populations. This is the area the only area that we found some carryover in timing. So let's delve a little bit deeper into how and where birds are utilizing that vast ocean basin on migration. This next paper was led by Nina O'Hanlon from the BTO and is a sister publication to the phenology study. We asked what areas were being utilized by birds during migration, and can that choice of staging area be explained by population? So you can see that the breeding areas of the subset that we used in this analysis, and the tracks are from around 131 individuals, with around half of them with multiple years of tracking. So firstly, let's focus on the migratory routes. You can see that there's a huge amount of variation captured there. And to account for the equinox effects, we used conductivity data to classify transit flights from stopovers in a two-state hidden Markov model to assess these key utilization areas from the daily behavioral budget. And that two-state model was verified from travel rates where positional data was available. So we found multiple discrete staging areas during both the south and northbound migrations in the North Atlantic, and this complex migratory strategy. So they're using multiple stops, and there's a huge amount of variation between individuals. We tested that migratory connectivity between populations, and again found a high degree of mixing, indicating that populations mixing occurs really soon after the start of migration and continues for the whole non-breeding period. And we tested that variation in strategy for either the duration or the proportion of migration of stopovers in breeding and wintering population strategies, and found significant interaction of the migratory period, so southbound or northbound, and the population. And this pattern was largely driven by that most northerly Svalbard population that spent less time in days in proportion of migration on stopover during that return and then on the autumn migration, and from other populations during that return migration. And this highlights the importance of staging areas during migration, and it may contribute to our understanding of this variation in population trends between these breeding sites throughout the North Atlantic. So this wealth of tracking data has given us a really rich framework to investigate the role of the migratory period in population processes, but there's still some open questions, and I wanted to dig a little bit deeper into how might the degree of connectivity between individuals across space and time together, and how full annual cycle strategies might drive population level trends. So as we know, seabirds are one of the most threatened groups of vertebrates. We can all imagine those trends, and almost half are experiencing some population declines. But we need to quantify this. So we're currently working to pull together the best available data for abundance throughout the range, and I wanted to generate a metric of connectivity to test how that temporal spatial organization of the annual cycle may drive population level processes. So I used a very work in progress. Using a graph theory approach, we can also quantify that network structure, and I'm using this approach to derive a single metric to parameterize population models and understand the interactions between complex movements and these demographic outcomes. And further to the understanding migratory connectivity and population dynamics, this body of work has the potential to inform conservation practices, disease transmission rates, and seasonal energetic budgets. But the proximate and ultimate drivers of migration are still unknown. So the patterns that we see may be driven by food availability, particularly at these staging areas and wintering sites, or perhaps it could be the cost of transport, especially when birds are undertaking these extremely long distance migrations. So this is just a first look at wind conditions across the range. Gives the impression that birds potentially take advantage of reduced cost of transport on long distance migration, and I'm currently formally testing this in a step selection function, but the idea seems to stand behind this survival analysis that we conducted in parallel. I'm going to quickly take you through that in the last minute, and life history trait theory indicates that long-lived vertebrates' adult survival is a major determinant of fitness, and therefore with population dynamics. And the environmental buffer hypothesis states that in long-lived species, adult survival is generally buffeted against these environmental conditions. So just to take from this slide, they breed up here, they winter down here. So we looked at environmental indices at these two very disparate regions of the globe and its effect on survival. And what we found very quickly is that in line with the environmental buffering hypothesis and the data set that we had using rings over 75 years at the Faroe Islands, the environmental conditions had no effect on survival, but we did see this really worrying downward trend in survival across the study period. And we also found some support for a similar downward trend in survival over time in young birds, but also an effect of the environment. And I see that I'm running out of time, so I just want to wrap up and thank all the funders for this. So come and catch me if you want to know more about these studies, and thanks to all the organisers, and particularly the Marie Curie Fellowship and the Centre for the Advanced Study of Collective Behaviour, and to you for your attention.
Okay, time for one quick question. Only one.
Thank you for your interesting talk. I will just want to say that we have observed some individuals near Cyprus in Levantine Sea during migration. And I wonder, because Levantine Sea is an oligotrophic area with not so many yellow-legged gulls, that if the birds there attack also to exhausted passerines during spring migration. And because we have observed these birds some years now, I mean continuously, do you have any idea also about the populations that those individuals may belong? Thank you.
Birds wintering in the Mediterranean Sea from the tracking studies, but it's really interesting that you highlight the importance of these heterospecific interactions between these kleptoparasitic species throughout their annual cycle. So their main resource is likely other birds. So what I really want to know is, where do all the other seabirds go? Because that's likely driving the processes underlying the migration of the Arctic skuas. Thank you.
Okay, thank you very much.
So during that period, I learned that it's really rare to see our study species drop dead from the sky. Or more in general, see adult seabirds die in their breeding colonies in large numbers. Well,
it's the better.
Okay, that's better. And yet, as most of you are aware of, this is exactly what happened from May 2022 onward, when highly pathogenic avian influenza, or bird flu, struck for the first time in the breeding season. And several seabird species were severely affected, amongst which sandwich terns. And by the end of the breeding season, it was clear that the impact on the population of these species in Belgium and the Netherlands was really severe. But how severe? And what was the impact on the demography of the species? And I'll try to shed some light on this during this talk. In 2022, we had 12 colonies with in total just over 18,000 breeding pairs in both countries. And all of these were affected by bird flu. And the impact was severe. Out of 36,000 breeding adult sandwich terns, 9,600 were found dead, or 27% of the population. And additionally, breeding success was really low, with chicks dying in their thousands, either because their parents were dead or just to infection with bird flu. And given the outcome of some virological models and the likelihood that many birds died away from the breeding colonies and went unrecorded, we feared that the actual mortality would be much higher. But how much? And what was the situation in other countries? And so to gain a comprehensive understanding of the impact across the entire Northwest European sandwich tern population, we reached out to site managers, to scientists, to ringers, to all people involved with sandwich tern colonies. We asked them to report the number of breeding pairs and the impact of bird flu on the colonies, if there was any. And so this collective effort provided us with a detailed picture of what happened to the European sandwich terns in 2022. The baseline situation we see here, with in total a little bit over 63,000 breeding pairs spread out over 67 colonies, and 39 of these were affected by the flu. The impact of the outbreak varied by country, with the most severely affected colonies located around the North Sea and in the Kattegat area. And initially, mostly adult birds died, with an estimated number of over 20,500 birds. And this represents about 17% of the total population. This would be a substantial loss for any species, but for a long-lived seabird that typically raises only one chick per year, this is quite dramatic. And the compilation of these data kept us warm during the wintering months, with anticipation for what the next breeding season would bring was high, because we were quite concerned, as alongside reports of outbreaks of bird flu in Western Africa, we also received many recoveries of ringed birds found dead on the wintering grounds. So, what did the 2023 breeding season bring us? Let's start with Belgium and the Netherlands, and for your reference, this was the situation in 2022. First of all, several large colonies that had been occupied for many years, sometimes dozens of years, and were severely impacted by the flu, in 2022 were abandoned. And this was not entirely unexpected, as sandwich terns often leave their colony site following a traumatic event, such as significant predation on adults or chicks, or high adult mortality in this case. So where did the birds go to? Most gathered in 4 large colonies. They did so in surprisingly large numbers. In total, over 14,000 pairs were counted, and although this still represents a decline of over 21%, this was much less severe than we feared, especially considering the fact that the year before, almost 27% of the breeding population died. And we could think of a number of possible explanations for this, of which relocation of birds from other countries to Belgium and the Netherlands would be one of the most logical. And to be able to test this hypothesis, we started again a round of data collection across Europe. So, how did the European population fare in 2023? For your reference again, this was the situation in 2022, with the affected colonies indicated by the many shades of red. In 2023, the total population in Northwestern Europe decreased by over 26% compared to the year before. And that's obviously a lot, but let's not forget that a minimum of 17% died the year before. On top of that, a little more than half of the colonies, but accounting for 90% of the total breeding population, were affected by bird flu again. And that being said, the impact of the epidemic differed markedly from the one the year before, because while adult mortality was again much higher than normal, with around 1,000 birds found dead, the percentage of adults that died was with 1% much less than in 2022. But it was the chicks that suffered the most, and strangely, in most colonies, they seemed to do very well during a large part of the chick rearing period. And then, when they were 3 weeks old or just before fledging, extreme mortality often kicked in. And this resulted in the second year in a row with a very low breeding success for a large part of the European breeding population. So when we look at the evolution in breeding bird numbers per country, we see some striking differences. We already saw that the decline of the Belgian and Dutch population was much less than the percentage of adults that died in 2022. However, Denmark and Germany experienced in both countries a 40% loss of the number of breeding pairs compared to the year before. And the UK and Ireland lost 25% of their breeding birds, with also many colonies that were not affected in 2022 showing a marked decline. Now it's tempting to think this suggests that a significant influx of birds from other countries to Belgium and the Netherlands might indeed explain the unexpectedly high numbers there. But these could equally be explained by relatively more young adult birds than normal starting to breed and filling the gaps left by the death of the many experienced breeders in 2022. The gaps could also have been filled by so-called floaters, birds that skipped a number of breeding seasons and started to breed again in 2023, and we could think of several more other hypotheses. So many questions remained unanswered, and a very important one for us was if the mortality was age-related. In other words, was the outbreak affecting primarily young birds, or were older, more experienced, but maybe also a bit more vulnerable birds being hit harder? So understanding this is crucial for assessing the species' future recovery. So we created the bazooka, a tool to aim at questions like this: an extensive data set containing all sandwich tern ringing data from the most severely affected countries from 1991 till now. And not just metal rings, we also included all long-term color ringing programs. And it's really extensive. After cleaning the data, we were left with over 100,000 unique recitings of nearly 170,000 ring birds. And based on these ringing data, we ran country-specific CGS models to estimate the base survival rate over 3 age classes: immature birds, so birds younger than 4 years, young adults 4 to 7 years, and older adults, which are birds over 7 years old. And by applying these survival estimates to the yearly number of ring birds per country, we could calculate the number of birds from each age cohort that were presumed alive in May 2022. So basically, the age composition of the ringed population before bird flu hit the colonies. And it might come as a surprise to some of you, but of the many tens of thousands of ring birds over the past decades, this is what remained just before the bird flu outbreak. And the first question we tried to tackle was if there is an age effect in bird flu-related mortality. So we first filtered the database for all dead recoveries of sandwich terns during the 2022 breeding season, most if not all birds that died due to the bird flu epidemic. And in total, almost 1,700 dead birds were found, of which over 1,400 ringed as 4 years in their first year. This is quite a lot compared to the average of 27 dead recoveries per year between 1991 and 2022. So when we look at the age composition of these dead birds, we observe a pattern that fits the expectations. We see that very few 2- and 3-year-olds were found dead, which is logical, as sandwich terns tend to stay in their wintering grounds in Africa in their first 3 years of life. We also see a high number of 4- to 15-year-olds and a decreasing amount of older birds. One has to take into account, though, that these graphs do not take into account the number of ring birds that were alive at the start of the epidemic. So after correcting for this, we see that there was indeed a very strong age effect of bird flu mortality on the European sandwich terns, with older birds being significantly more likely to die than younger ones. And this age effect also remains consistent across the individual countries. So what does this mean for the age structure of the European sandwich tern population? We clearly see that especially the older birds and the most mature birds, that this group is beheaded on the European mainland. In Belgium and the Netherlands, we lost around a third of all birds older than 7 years. And of the birds older than 20 years, we lost around 75%. So now what? Luckily, bird flu was absent during the breeding season of 2024, and good for the birds after 2 years in which they lost more than a quarter of their population, a huge chunk of the most experienced and mature birds, had very bad breeding success, and good for us as now we have time to aim our bazooka at questions related to the composition of the new population. And in the end, we will include all this in the population model to assess the species recovery potential. In the meantime, let's hope bird flu does not return in Europe, also disappears in the rest of the world. So pictures like these are a thing from a dark past, and we all know that a lot of our seabirds can use this. Thank you.
Thank you very much. Questions for Wouter? There's one down here. Adriana.
Fantastic study. Thank you for this talk. Do you have an idea or a guess of what proportion of birds may have been infected and survived? And whether this could change with age?
That's a bit hard to say at the moment. We know of birds that were infected and survived, but probably, especially in 2022, I think most birds that were infected died. And in 2023 we had another situation, but the virus changed a bit, the genome of the virus changed a bit. And apparently, because we saw a huge infection in the young sandwich terns, they all died. Parents tend to stay alive, although some of them we know were infected. So it depends between years, at least.
So it could not be that some got infected the first year and developed antibodies and resisted the second year? Like the, well, not the young ones, but...
I didn't get the
sorry.
sorry.
Depending on how many got infected and survived, those that got infected and survived may be immune to the virus.
We know there's quite a lot of immunity as well.
Mm-hmm.
We tested, I think, almost 200 adult birds in 2023, and depending on the colony, and it seems a bit latitude related, we saw between 25 and 56% immunity.
Okay, thanks.
Or antibodies.
Yep.
Immunity. Yep.
Thanks.
Okay, another question? There's one here. Adriana? Aqui embaixo, aqui.
Thank you. Hi. For those now ringing to replace in the ringed population the same number of birds that kind of died in the avian flu outbreak, would you ask them to collect any other data whilst they're ringing at the moment, which would be useful for you in the aftermath of the next avian flu outbreak?
Sorry. Can you repeat, please?
So there will be lots of ringing effort going on at the moment to replace the birds who died in terms of monitoring effort. There's likely to be another outbreak of avian flu in the future. Would you ask those ringers to collect any specific data before that happens now, whilst they're doing that ringing?
And keep on reading rings. Yeah. Yeah. Especially color rings.
Okay, thank you, Walter. Thank you. Now, Ingrid. She's going to talk about adult survival in Leach's storm-petrel. Up and down, and yeah.
Perfect. Try to sit on that there.
Okay, the microphone is on.
Can we up or down?
Thank you, everybody, for being here. Before I start my talk, I'd like to say that the results I'm going to present today are not published yet. So please refrain from posting any result slide on social medias. Any other slide is totally fine. So I know this is a pretty young crowd, but how many of you were at the Edinburgh 2016 conference? Okay. For those who were not there, there was this sentence that became somewhat the motto of that conference, and it's never too late to start a long-term study. So while I agree that it's never too late to start a long-term study, I started one myself in 2010, I think it's also important that you have to start analyzing the data from those long-term studies. And as a case study, let me introduce you to Chuck Huntington. So Chuck was a professor at Baldwin College in Maine. He became the director of Kent Island Field Station in 1953. And Kent Island has a decent-sized Leach's storm-petrel colony, so in 1955, he started a study on them. He probably didn't know at the time that it was going to become a very long study, because it's still ongoing 70 years later. But Chuck passed away in January 2017 at the age of 98. He was still going to Kent Island every year, but there's very little data published on all of his work. So what kind of long-term study are we talking about? For every year since 1955, except for 1977, when Chuck was on sabbatical and nobody was on the island. Active burrow and selecting plots were monitored. Adult Leach's storm-petrel were banded. Morphometric measurements were taken. Egg lay date and hatching success were recorded. So as you can imagine, there's a lot that can be done with all that data. And a while ago, I was given the opportunity and the immense privilege to work with that data set. So the first thing I wanted to tackle was an adult survival analysis, because Leach's storm-petrel population is declining in Atlantic, and we are trying to understand the cause of this decline, and low survival could be a possible cause. So the basic of survival analysis using recapture data is pretty simple. It's based on two different probabilities: phi, the apparent survival probability from 1 year to the next, and p, the encounter probability on a given year. It's not because the bird is alive that you're necessarily going to see it. To have a robust survival analysis, you need usually at least 5 years worth of data. So I think we were pretty good on that front. But of course, it's more complicated than that, otherwise it wouldn't be fun. Each of those variables can be constant, can vary over time, can be site-specific, can be dependent on the time since marking, and any of those combinations. So in this case, we didn't want to simply determine the apparent survival rate, but also the potential effect on environmental variable on the survival. So what we first did is determine the best model, and then we used this model as a template and added different environmental variable. And which environmental variable did we pick? Well, we picked the North Atlantic Oscillation, which is the pressure difference between Iceland and the Azores. I think by now you know about this. There's strong positive phase, and it relates to abnormal temperature in eastern US and northern Europe. And we can observe long period of either positive or negative value. And there's often a time lag between the North Atlantic Oscillation and biological responses. So we use yearly, summer, winter values, 1 year lag, 2 year lag. So right there we already had a lot of different variables. Then we use the Atlantic Multidecadal Oscillation. It's based on variation in sea surface temperature in the North Atlantic Basin. The estimated cycle time is 60 to 80 years. And keep in mind that it's currently at its high point. It might come in handy later on. So with the North Atlantic and the Atlantic Multidecadal Oscillation, we have somewhat regional indices confined to the North Atlantic. So then we pick a more global variable, which is the annual global mean temperature anomaly. And it's derived from both marine and terrestrial temperature, and it's been on the rise since the 1950s. And on Kent Island, we noticed that the annual global mean temperature explained the variation of the reproductive success of Leach's storm-petrel. So we thought we should probably add it, because if we explain the reproductive success, it might also explain the survival. And then finally, we picked the Southern Oscillation Index. You've heard about it a lot. And it's like, well, why picking a Southern Oscillation for like birds breeding in the north? Well, they also travel in the south. And positive and negative phases of the Southern Oscillation has consequences reaching further than the Pacific Ocean. And here's a graph of the probability of survival of blue petrel against the winter Southern Oscillation Index with a 4-year and 3-year gap. So we also use different gap for this index. So Kent Island is located in the Gramenan archipelago at the entrance of the Bay of Fundy. 3 plots were started in 1955 by Chuck. But because of the change of vegetation and the inaccessibility of most of those burrows, the plots were faded out in 2001, in 2007, and a 4th plot was created in 2003, and it's still active to this day. In total, there were over 7,000 individual banded and almost 25,000 recaptured. So on this graph, for each year, you have, and each site, you have the number of new banded bird in light gray and the number of recaptured in dark gray. The best model for the survival showed a time and side effect for the survival rate, and the recapture probability vary with time and a site interaction. So here's the recapture rate probability. Overall, it was 0.2. And as you can see, the recapture probability was fixed to 0 in 1977. As I mentioned, there was no one there. And in 2020 and 2021, there was COVID years, and work on the island was limited. So at best of time, only half of the birds could be determine like who was in the burrow. So for the apparent survival, the survival rate overall was 0.8, which is not that bad when you think about it, but then it varies a lot from 0.6 to 0.97. And a lot of the time you hear that apparent survival in Seabird should be around 0.9 or greater to maintain population stability. So here is the 0.9 mark, and as you can see, apparent survival was above that on only a very few years. So now if we look at the survival and the different environmental covariate, we see that the Atlantic Multidecadal Oscillation was the best environmental variable to explain the apparent survival rate, but it only explained 10% of the variation in the survival. Remember that we are currently at the peak of the Atlantic Multidecadal Oscillation, so hopefully that will provide better condition in the future year for Leach storm-petrel. But we have to keep that with a grain of salt because he only explained 10% of the variation. So if environmental variable only explain a fraction of the variation in survival, what other factors could explain the low survival? Well, as I mentioned earlier, population of Leach storm-petrel is going down, and several teams are currently working at different colonies across the Atlantic Canada to try to understand the cause of the population decline. So using up to 20 years of marked recapture data, we recently estimated the survival at 6 colonies, including Kent Island, 2 colonies in Nova Scotia, and 3 colonies in Newfoundland. So the survival was lower, it's like the blue line. It was lower at colony with strong predation pressure, and that predation mostly came from gull, but also survival had a tendency to decline across colony with a high mercury burden. So on this graph, each colony is represented by 1 point. So in conclusion, we can say that the Atlantic Multidecadal Oscillation, it's a mouthful for me, was the best environmental variable tested to explain the variation of survival in Leach storm-petrel on Kent Island, but only explained 10% of the variation. This weak trend could not have been detected without long-term data set, as the Atlantic Multidecadal Oscillation has a cycle of 60 to 80 years. So it's important to maintain long-term database, even if funding can be difficult to obtain for such data to be collected. And as I mentioned earlier, it's just as important to analyze the data. So there's a lot more that can be done with this data set. I'll probably see you in 2027 to talk about pair bond and divorce rate in this species. And there's other factors influencing the survival of this declining seabird. And for more about this, I think you should not miss April's talk this afternoon at 2:30. So drink some coffee and stay awake. Finally, I would like to give a huge thank you to the different directors of Kent Island Research Station, including Bob, Mark, and Patty Jones that give me the opportunity to work on this data set, to all the students that collected the data, and thank you for listening.
Thank you, Ingrid. Questions? Adriana, down here, please. Just put these off when they talk.
Thanks, Ingrid. It was really great to see those results and fascinating talk. I was really curious about the underlying population trends of Kent Island, and in particular, is it stable? And is it, in your opinion, a saturated population, or has it ever been in the past a saturated population?
In terms like if there's not enough habitat for them?
Yeah.
No, it's lots of place.
And the population trends over that?
The
time
population is declining.
period?
It's been declining by about 30% in the last 30 years.
Okay, I think.
Hello everyone, can you hear me? Yes, good. My name's Eve. I'm a PhD student at the University of Liverpool, and I'm going to present some of the preliminary results from my second chapter, which is looking at quantifying the responses of UK seabirds to extrinsic threat using PVA. And I apologise in advance for the lack of pictures of seabirds in this talk. So as everyone in this room knows, seabirds are affected by multiple different human activities, and each of these activities can exert a different range of pressures on them. And some of these pressures can be direct, like collision mortality or bycatch mortality, which we've talked about, and some of them are indirect, such as the effects of fisheries competition and some of the effects that we're seeing of climate change. And there are multiple sensitivity assessment frameworks for looking at how these pressures affect seabirds. But most of them are underpinned by kind of qualitative estimates of sensitivity, and it's quite hard for them to trace population consequences all the way to specific combinations of activity and pressure. So what my project aims to do is to use PVA in order to explore the population consequences of a wide range of demographic impacts, in order to create a quantitative foundation for the broader assessment framework that I'm trying to make throughout my PhD. And the hope is that if we model a crazy range of demographic impacts and how they affect population consequences, we might then be able to map some of those consequences to the specific activities that are causing them. And this will allow us to compare different activities on a common scale, and therefore hopefully aid us when we're trying to make prioritization decisions in conservation. So briefly, population viability analysis, or PVA, is a method of predicting how populations are going to change in response to changes to their demographic rates. And it's often used in environmental impact assessments to look at how a seabird population might respond to a specific industrial development. And Natural England in the UK have developed a specific seabird PVA tool, the underlying R code and package for which is what I use for PVA in my project. So yeah, I wanted to use PVA to look at how key species are going to respond to impacted demographic rates, how this varies between species, how it's affected by the addition of density-dependent regulation, and how these things interact with impact severity and impact duration. So firstly, I chose 5 species, broadly representative of a range of UK seabird demography and life history strategies. I went for fulmar, guillemot, shag, gannet, and kittiwake. I'm actually not going to talk about that. And yeah, and then, so I chose a range of different impact scenarios, a massive range, and I included some positive ones in there as well so that we could look at how conservation outcomes might have positive effects. And I based it on a broad literature search of kind of different quantified impacts to population demographies by specific activities. And then so I've got survival scenarios, I've got breeding success scenarios, I'm timesing them by each other. I've got 5 different scenarios of impact duration, and then I've got my different density dependent scenarios, and this all adds up to about 30,000 PVA runs per species, which, as you can imagine, is a massive computational headache and has created a deluge of results which I'm still trying to untangle, but I'm going to try and show you some examples and hopefully extract some headlines. So these are my predicted trajectories for each of my 5 populations over 25 years. This is with no impact. It's just using national demographic rate averages for breeding success and survival over the UK. And as you can see, they kind of look broadly reasonable, apart from the guillemots, which are absolutely taking off. And I think that that is a consequence of using a density-independent model to model a population that's much more likely to be under negative regulation. And I'll talk more about that later. So yeah, some impacts. These heat maps show broadly the proportion of the population that remains after 25 years of different impact scenarios. So each of these mini plots has a different impact duration, but they're all being measured at the end of 25 years. And I've got 1 year, 3 years, 5, 10, and 25 years. And the darker the colour, the more severe the population decline. So the kind of lighter colours are population increase. And it's going from positive to negative, and it's the other way, isn't it? Positive to negative. And I've put some thresholds in there as well, just to kind of aid interpretation. So if you're to the right of these kind of the black box in the black box, then that's any kind of population decline. The pink line is indicating 80% proportion remaining, and then we've got red at 50%. And if you're within the white box, then there's less than 10% of your population remaining after 25 years. So yeah, for example, we can see here that guillemots do much better overall in density-independent Sept scenarios than Fulmar and gannet. They're much less sensitive to those shorter durations. So we're only getting the kind of 50% loss when our impact's persisting for more than 10 years. Whereas Fulmar seem to be very sensitive to even 1 year of decline, particularly in breeding success. But as we'd expect, they're different species, they're differing in their responses to a wide range of impact scenarios. But how does this change when we add density dependence? So briefly, density dependence is when a population's vital rates, for example, breeding success and survival, are regulated by its size or density. So this relationship can be negative or compensatory, which is, for example, if having a very crowded population actually reduces individual breeding success because of that overcrowding. Or it can be positive or depensatory, and this is where you might find that population loss actually reduces survival for individuals because you're losing that aggregation defense against predation. And it's not often included in PVA-based impact assessments because we assume that it will be compensatory, and therefore that not including it provides us with a precautionary assessment. And you might have already heard me bang on about this in Cork, but I found in my previous study that we found examples of positive and negative density dependence in various populations across the UK. I found it in kittiwakes, guillemots, shag and fulmar, so 4 of my 5 species. And so we decided that for this project we'd run all 3 scenarios, so positive, negative and density independent for all of my PVAs. And I've used the coefficients that I generated in this chapter 1 to kind of describe these relationships through this process. So now if we look at guillemot change in response to impact, we can see that under a positive regulatory scenario, under short impact durations, that guillemot population is increasing even more with the positive impact scenarios. Whereas under negative regulation, we're finding that, which is a much more probable scenario for a thriving guillemot colony, we're finding the populations remain stable over 25 years when impact duration is short, and that they're experiencing less extreme declines as well, even at longer impact durations. Finally, some pressures, like oil spills, can happen in a moment and cause extraordinary amounts of damage, whereas others, like mercury poisoning, which we just heard about, can chip away at survival for barely noticeable increments over decades. And we call these acute and chronic impacts. And so my third objective was to look at how impact duration interacted with degree of impact to affect population outcomes across different species. So here we tried to visualize that. We're looking at impact duration, strength of impact, and survival versus productivity. And I don't really have time to go into it, but hopefully you can see that in our 2 increasing species, so the gannet and the guillemot, there's a lot more variation in their response to these low versus high and productivity versus survival changes than there is in the 3 declining populations. And I think in an density independent scenario, a lot of species are showing a kind of equivalent between that 1 year acute impact and a 25-year kind of longer chronic impact. Whereas when we're looking at, for instance, the negative density dependent scenario, it's a lot more consistent and less dramatic change below 25 years of impact where it starts to change again. So in conclusion, different species respond differently to different impact scenarios. Some seem to be more sensitive than others to impact severity and to the duration of impact and to the addition of different forms of density dependence. And that seems like a very obvious conclusion. But lots of the impact assessments use PVA that doesn't necessarily account for a lot of these different factors, so it's important that we explore it. And even though we all know that seabirds are vulnerable, declining, and that the picture of the future that we have often looks very bleak, my case funders assure me that sometimes we need to be able to prove that. So yeah, my hope is that the way this can be used in my framework, in my third chapter, is that a practitioner could kind of take an estimate based on expertise or published evidence of the specific demographic impact of a particular activity, and then they could use kind of some of these model outcomes to predict what a range of population outcomes for that activity could be. And then hopefully use those density dependence, positive and negative scenarios as a kind of best and worst case scenarios, so you've got a proper kind of degree of uncertainty as well. And that's the approach I'll be developing more in my final chapter, which I'm hoping to call on some of you for your expertise in. Finally, I put up this very overused quote, because I think it's true, which is that all models are wrong, but some are useful. And I think that from the outside, population modelling can seem like a mysterious window into the future. But once you're doing it, you realise that it's incredibly messy and sensitive. And for my brief stint working with PVA, I think that it can be a really useful tool as part of a holistic decision-making process and as a comparative tool. But I don't think that it should be dictating decisions about the future of seabird communities or any other more than human communities. Thank you very much.
Okay, time for questions. Martin, just put this off.
Yeah, very nice talk. Perhaps I missed it about what kind of assumption did you do with the increased mortality in relation to impact? So you used the model to see what the impact is on the population, but what kind of assumption did you do in relation to mortality, increased mortality?
Assumptions in relation to what? Assumptions underlying the modelling of mortality specifically.
Oops. Yeah. Well, perhaps you can speak later then. Yeah, yeah.
Okay. Another question, not on assumptions. No, I'm sorry.
Not on modelling.
Thank you for your talk. I was wondering, have you tried to compare your model with the data that we can have, like between, for example, 2000 and 2020, to see the accuracy of your model, and maybe it can help you to identify some links between the seabirds and some parameter variables?
Yeah, so these are very much theoretical populations, and they're based on UK demographic rates. But for that previous work that I talked about, where I talked about density dependence, I did an observational analysis where I ran lots of different seabird populations using demographic data for populations in the UK, and I compared how different models would recreate those observed dynamics in those populations, and found that it's very varied whether which density-dependent model you use, how accurately they'll create observed dynamics. But actually, often when we're looking at impact assessments, we're not necessarily, well, not we are, but the impact assessments aren't always using models that are trying to recreate observed dynamics, because they're often looking at that comparative between a baseline trajectory and an impacted trajectory. So sometimes the model that creates the most accurate recreation of an observed dynamic actually also gives you, I guess, the least precautionary assessment of an impact. Does that make sense? So yeah, it is really interesting.
Okay, thank you.
Thank you.
Now we have a round of the speed talks, 1, 2, 3, 4 speed talks. We start with Roger. Foraging strategies in anthropogenic landscapes modulate pathogen load and antioxidant status in an opportunistic predator, which is a yellow-legged gull, I believe. Okay.
And then for the point,
Yeah.
Yeah, yeah.
Ah, here.
It's here, yeah.
Okay, perfect.
Yep.
Okay, so thanks everyone. I'm going to talk about how foraging in anthropogenic ecosystems affects antioxidants and pathogens in seabirds. So as you know, foraging strategies are associated with physiology. You can see here how diet and foraging habitat has been associated in penguins with oxidative damage and also antioxidants. But it's not just foraging, it's also some features of this foraging. You can see here on your right graph how longer foraging trips are associated with higher levels of oxidative damage, and those higher levels, yeah, you can see that. Higher levels of oxidative damage, they are associated with lower energy expenditure in terms of bedba. So it seems that seabirds, when they forage, they optimize their foraging. And yeah, we can think about that this optimization of foraging is like a balance between the energy that they use and the energy that they get from the diet. So when we are talking about seabirds that they are opportunistic foragers, like the yellow-lag gulls, they forage in this kind of ecosystems where you have natural areas, but also urban areas, areas with factories, and also agricultural land farms. So this can disrupt the balance in foraging of these seabirds, and they might forage with resources that they are infected, and they can increase their pathogens, and finally they can disrupt their physiological status. So we wanted, what we wanted to ask here is whether this foraging in different periods of time is affecting the antioxidants and also the zoonotic bacteria in the seagulls. So to do this, we sampled 30 yellow-leg gulls in southern Spain in 2015. We also took stable isotopes, and we assessed stable isotopes in 3 different tissues that they were actually representing 3 different periods: incubation, chick rearing, and wintering. From here, we studied diet and foraging habitats, but also we assessed the percentages of foraging resources, whether they were pelagic, demersal, or terrestrial. And finally, we assessed antioxidants and 3 zoonotic bacteria. So moving to the results, first of all, what we saw is that gulls that they were foraging in demersal resources in inshore locations, they were associated with higher prevalence of chlamydia. But this was only during incubation. For the rest of periods and the rest of pathogens, we didn't find any relationship. And moving now to the antioxidants, during incubation, those gulls that they were foraging in pelagic resources in inshore locations, they present higher levels of glutathione peroxidase and lower levels of glutathione, which are antioxidants. And this was during incubation. And during chick rearing, also higher levels of glutathione peroxidase were associated, or were found in those gulls that they were foraging in low trophic level praise, pelagic resources, and inshore locations. And finally, also during chick rearing as well, gulls that they were exploiting demersal resources in inshore locations, they were associated with higher levels of TBERS, which is another antioxidant. So just as a take-home message, the foraging habitat of gulls or seabirds in general can be associated during breeding to zoonotic pathogens, and also depending where seabirds are foraging and the diet that they have during chick rearing and incubation, and also as in terms of trophic resources, is associated with a different antioxidant status. And finally, we did not find any association during the wintering period. And thanks for listening.
Now is Anne-Sophie from La Rochelle. She's going to talk about subtropical brown skuas.
Hi. So as Maria reminded us, invasive alien species are one of the main threats to seabirds. And in response to that, a number of eradication programs have been set up in the hope that eradicating the invasive alien species will lead to beneficial effects for the locally breeding seabird populations. However, these programs can sometimes have some detrimental effect for the local species. So in this context, Amsterdam Island was identified as a hotspot for seabird diversity and was also identified as a priority site for the eradication of invasive alien species. So in response to that, an eradication program was planned. However, there are concerns that this program might have some detrimental effects on the local population of subtropical brown skuas. And this can be either through secondary poisoning or through depletion of the prey base. And why bother about this population? Well, first, this population is rather small, around 80 pairs, and also some recent genetic results by Laura Martinez-Anton and Amelia Viris-Elpon showed that this population is actually quite strongly segregated from other populations of the same species in the Indian Ocean. And it's about as differentiated from these populations as it is from another species of skua, the South Polar skua in Adélie Land. They also found that the genetic diversity of the Amsterdam population was lower than the diversity of other populations, and they found a much lower effective population size than in other populations, and a high rate of private alleles for this population. So this suggests that there is a diversity that needs to be protected. So in this context, there was a need for some ecological information on this population prior to the eradication. So for this, we collected some GLS tracking data in order to better understand the migratory behavior of the seabirds, of this species, and also in order to try to identify an optimal eradication period, which would be the period where, when we would have the lowest amount of skuas on the island. So here you see the non-breeding distribution of the skuas from Amsterdam Island. A non-breeding distribution that is actually quite similar from what was known from other populations, so populations from Crozet and Kerguelen. We see a quite high inter-individual variability in the destinations that the skuas have reached, and also in distance, and I'm not showing this here, but also in activity budgets throughout the year. This high inter-individual variability comes along with some actually high individual consistency in the routes and the destinations that the skuas take. And this inter-individual variability is actually quite extreme, going from some long-distance migrants to some birds that are actually fully resident. So if you look here so in orange, you have for each bird the time spent at the colony, and in blue the time that is spent at sea. And you can see that 2 of the tracked birds actually remained at the colony all year round. And this is actually reflected in the immersion data, with these resident birds actually barely spending any time on the sea surface throughout the year. So this tracking data allowed us to identify what would be a best period for the eradication that would go from May to end of July. And in response to, following these recommendations, but also some practical considerations, the eradication program started this year in June. So in conclusion, the population from Amsterdam Island constitutes a distinct evolutionary unit that is vulnerable given its low population size. And we found some high variability in migratory strategies, including some birds that were fully resident. And we don't know what these birds feed on yet, but there are some ongoing isotopic analyses to try to get a bit more information. And this allowed us to determine an optimal eradication period, but this still raises the question of what can happen to these birds that stay year-round on the island. Thank you.
So finally, Antoine is going to talk about social interactions beyond near neighbors in Atlantic puffins.
Okay, can you hear me? Yeah, perfect. My name is Antoine. I'm from the University of Merrall in Newfoundland, Canada. And for my PhD research, I'm mainly investigating the factors that influence social network on Atlantic puffin. And this talk is about to present some of those results. So when we talk about social network, we acknowledge that association mainly occur between individuals that have similar needs that match temporally and spatially. We also understand that spatial environments, so everything related to distance between burrows or territories or individuals, and social environment, which is more group structure, for example, all of this can really affect social network. What we don't know is how those mechanisms can contribute to changing social network on seabird. If you look at Atlantic puffin, well, they breed in very dense colony, we all know that, but they still have their own burrows, they still have their own territory, and they can move around. So basically, if they want to see someone, they can, so that they just have to move and go. And this really opens the door of the social environment. On the top of it, so Atlantic puffin have a high breeding philopatry. They're also long-life seabirds, so we can expect from them to understand or to have a good understanding of their local neighbor and local environment. That's why we decided to use Atlantic puffin to study spatial and social environment and those effects on social network. So for this, we pick a site on the east coast of Canada in Newfoundland. It's the biggest colony of Atlantic puffin in North America. And we trapped and banded at night 125 individuals with color band. We also took GPS coordinate of their burrow, and we performed some focal observation from a blind. So here we decided to, an association for this study is defined as any individual that is within a radius of 2 meters from a focal individual. And in terms of statistic, we look at the influence of burrow distance on community structure using a randomization test. And we also look at the frequency of association for all individuals within the same radius distance. For some results, so we found a strong effect of spatial environment on the social network. If you look on the right of this figure, the left, you have a social network graph, and each color represents a cluster. If you transpose those clusters on a map, you will realize that the clusters are also spatially distributed. The fun part of this is that pairs are not always in the same cluster, and the small anecdote is that when it happens, it's only the females that are not spatially distributed. For the social aspect, we couldn't find any effect of the social environment on social network. If you look at this figure, you will see a TOU value. So basically a TOU value, it's the equivalent of a correlation coefficient between predicted and observed value. So it basically shows how well theoretical value can explain the observed value. So when it's close to 1, all the associations are random. If it's not close to 1, the association may not be random. So from 2 to 16 meters, you can see that there is some favored association between individuals that nest far away from each other. So basically, we found a strong effect of spatial environment on social network. This probably comes from puffin that when they are on land, well, they don't move that much. They stay in front of their burrows, they protect their territory. So basically, the associations they have with others is just individuals that have the same behaviors than them. They just stay in front of their burrow and they protect their territories. They do the same thing. So this is the first thing. We couldn't find an effect of social environment on the social network. This is because all the associations were random, so they don't care, basically. We don't know if they can recognize each other, but what we know is those kind of associations mainly occur between individual or like species that use many eyes effect, or it's just an anti-predator behavior. They just want to be all together. They don't care about with who they are. We found some favored association between individuals that nest at long distance, but this remain pretty anecdotal. Can come from individuals that relocate their burrow from a year to another. So as a conclusion, Atlantic puffin clearly meet opportunistically with conspecifics that match temporally and spatially with the same needs. That is an anti-predator behavior. In some rare cases, they can have some favored association, but this remains really anecdotal. And for the next step would be to use interaction instead of association, so to define what kind of interaction we are talking about, and this could clearly open the new field of multi-layer social network, but this is way more complex. I would like to thank you, well, everyone in the project, my sponsor, but as well Pierre-Paul Bitton, my supervisor. Thank you.